session is always led by the mace bearer, the Deputy Registrar Senate Affairs following, and leading our deans and directors, deans over 13 faculties and counting, and then principal officers of this university. It will interest you that there is one particular individual who is going to be a part of the inaugural lecture in her new role and capacity. Ladies and gentlemen, please rise to receive the academic procession. Our deans and directors, all of whom but a few have all stood on this stage to deliver their inaugural lectures. And so, like a cloud of witnesses, they are here to chair Professor Ekundayo, the 34th inaugural lecturer, and that's the registrar, the deputy vice chancellor administration, the deputy vice chancellor academic, the inaugural lecturer, and of course, the vice chancellor of our university. Ladies and gentlemen, while we remain standing, we will take the national anthem and the Federal University Lokoja anthem. The national anthem, please.
Tribe and tongue made different, in brotherhood we stand. My gentle and proud to serve our sovereign motherland.
The university anthem.
Greatest FUL.
You may be seated, please. The Vice Chancellor of the Federal University Lokoja, officers of the university, very distinguished members of Senate, provosts, deans, and directors, heads of departments and units, very senior members of the teaching and non-teaching staff of the university, esteemed guests, gentlemen of the press, and our very excellent students, ladies and gentlemen, welcome to the 34th inaugural lecture of the Federal University Lokoja, to be delivered by Professor Emmanuel Olufemi Ekundayo. It's my pleasure, if you clap your hands right now, to invite very respectfully the registrar to begin formally the procession of this inaugural lecture. Another round of applause as he comes to the microphone, please.
Distinguished ladies and gentlemen, you're welcome to the 34th inaugural lectures of our great university, to be delivered by Professor Emmanuel Olufemi Ekundayo, titled Biotechnology, the Science, the Products, and the Business. Now, at this junction, let me humbly invite our Vice Chancellor, Professor Olayemi Akinwumi, to welcome all our guests and also introduce our inaugural lecturer and declare this inaugural lectures open. Vice Chancellor, sir.
Thank you, the acting registrar, the Deputy Vice Chancellor Academic, the Deputy Vice Chancellor Administration. This is your first official inaugural lecture. Please a round of applause for the Registrar and Secretary to Council, the Bursar, the University Librarian, Provost, Deans, Directors, and Head of Department, our inaugural lecturer, Professor Emmanuel Olufemi Ekundayo, members of the University Senate, academic and non-academic staff, members of the Ekundayo's family, student friends, and distinguished guests. And just on a lighter mood, just last week, new chief of staffs, service chief were appointed. I like the army tradition or the military tradition. Immediately, the president decorated the chief of staffs. They turned backward and salute their wife. My wife is here. Good evening. Ladies and gentlemen, I'm delighted to welcome you all to the 34th inaugural lecture of our university. We thank God for bringing us this far and for granting us the grace to witness yet another successful inaugural lecture, a cherished tradition in our academic calendar. The inaugural lecture, as many of us know, is not just an ordinary academic ritual. It's a deeply symbolic and historic academic event in university. It is a platform through which a scholar formally presents to the academic community and the public his or her journey in research, teaching, and service. It provides an opportunity for the audience to gain insight into the lecturer's field of study, their contributions to knowledge, and how such work impacts society. At Federal University Lokoja, we consider inaugural lecture series as one of the important avenues through which we project the intellectual wealth of our staff to the nation and the world. It is also a means of boosting research activities within our university, stimulating collaboration, innovation, and creativity. Through this platform, we have continued to strengthen our reputation as a growing center of academic excellence and a contributor to national development. Today, we are privileged to have one of our distinguished scholars, Professor Emmanuel Olufemi Ekundayo, a professor of biotechnology, deliver this 34th inaugural lecture. His lecture titled Biotechnology, the Service, the Product, and the Business, Harnessing Indigenous Bioresources for National Bioeconomy, is both timely and thought-provoking. This lecture marks a historic moment for the Department of Biotechnology, as Professor Ekundayo becomes the first academic from the department first academic from the department to deliver an inaugural lecture. In so doing, he's opening the door for others to follow, thereby setting a worthy precedent for his colleagues and younger academics in the discipline. On behalf of the management, senate, and entire university community, I wish to heartily congratulate Professor Emmanuel Olufemi Ekundayo for this bold step, a step that marks the fulfillment of an important and mandatory academic obligation. I also wish to specially recognize and appreciate the family of Professor Ekundayo for their support, encouragement, and sacrifice throughout his academic journey. I guess the wife and the daughter is here. Children, please, can you stand up for recognition? The family, please. Yeah, please, a round of applause for them. Thank you. Thank you, madam. Thank you, children. Yeah. Indeed, his success and achievement today are not unconnected to your love, prayers, and understanding. You share in this glory, and we celebrate you as well. Let me also extend my sincere appreciation to the inaugural lecture committee and all individuals and units that have worked tirelessly to ensure the success of this event. And let me also specially recognize Professor Salau, the last inaugural lecture. I can see her sitting down here. We are also getting old in the business now. As we listen to today's inaugural lecture, I have no doubt that we will all be enlightened, inspired, and motivated to appreciate even more the power of research and innovation in driving national development. Once again, I welcome everyone to this memorable occasion and thank you for being part of this celebration of scholarship. Thank you and God bless you all.
The Vice Chancellor, sir, may I humbly request you, sir, to read the citation of the inaugural lecture.
Prof, can you step forward here, sir? Citation on Professor Emmanuel Olufemi Ekundayo. Professor Emmanuel Olufemi Ekundayo was born on February 4, 1965 in Oduara, Kogi State, Nigeria. He began his educational journey at Oyi Local Government Management Board, Oduara, in 1974. Thereafter, he proceeded to Federal Government College, Ilorin. Federal Government College, Ilorin. I was there for my HSC, 1980, Charity House, Unity House. Wow. So we are just doing that. Where he distinguished himself academically. A recipient of the Federal Government Scholarship, 1981 to 85, he completed his secondary education with excellent results in the West African Examination Council, WAEC, GC O-level in 1985. Despite gaining admission to University of Nigeria, Nsukka, in 1986, financial constraints delayed his university education. However, undeterred, he secured admission to University of Jos in 1987, where he obtained a BSc second-class upper division in zoology in 1991 and MSc in medical microbiology from the same university in 1994. Between 1995 and 1997, Professor Ekundayo attended the Federal School of Medical Laboratory Technology as a graduate intern, where he obtained the Associate of Institute of Medical Laboratory Science with a specialization in chemical pathology. Professional career and experience. Professor Ekundayo began his professional career as a research assistant with the Herbal Medicine Research Group at University of Jos. In 1997, he joined the International Center for Ethnomedicine and Drug Development, Interseed, Nsukka, as Bioresources manager and microbiologist under a collaborative arrangement between the Herbal Medicine Research Group and Interseed. His work involved the antibacterial screening of medicinal plants, contributing significantly to ethnomedicinal research in Nigeria. In 1999, he transitioned to the private sector as application specialist and pioneer branch manager of Dalex Nigeria Limited in Port Harcourt. In this role, he received advanced training both locally and internationally on immune assay system and trained numerous medical laboratory scientists across tertiary hospitals, oil company, clinics, and government research institute. Driven by an enduring passion for academics, Professor Ekundayo joined the Michael Okpara University of Agriculture, Umudike, in 2005 as an assistant lecturer in the Department of Microbiology. He obtained his PhD in medical microbiology from the same university and rose steadily through the ranks to become a professor of medical microbiology in 2020. Administrative and leadership roles. Professor Ekundayo's leadership capabilities were evident early in his career. He was appointed acting director of the Center for Molecular Biosciences and Biotechnology, CMBB, in 2015. While still a senior lecturer, his transformative leadership led to his reappointment for a second tenure by a succeeding vice chancellor. Within the university, he also served in several capacities, including research program leader, biotechnology and genetic engineering research group, chairman University Laboratory Equipment Installation and Maintenance Committee, member University Tender Board, member University ICT Committee, member University Ceremonial Committee, professional affiliation and service to the community. Beyond the university environment, Professor Ekundayo has made remarkable contributions nationally and internationally. He served 2 terms as Vice President of the Biotechnology Society of Nigeria, BSN, and is an editorial board member of Umudike Journal of Engineering and Technology. He is a respected member of several professional bodies, including Biotechnology Society of Nigeria, Nigeria Society for Microbiology, American Society for Microbiology, Nigerian Genomics and Bioinformatics Network, International Union Against Tuberculosis and Lung Diseases, Association of Medical Laboratory Scientists of Nigeria, Guides of Medical Laboratory Doctors, International Network of Government Science Advisors. Over the years, Professor Ekundayo research has focused on harnessing indigenous knowledge and bioresources to combat infectious diseases of public health importance. His work encompasses gathering data on antimicrobial resistance, evaluating diagnostic tests for the tuberculosis, TB, exploring the antibacterial property of medicinal plants used in traditional medicine. He has published and co-authored about 50 peer-reviewed journal articles and contributed 2 chapters to 6-volume book series. Multidisciplinary applications in biotechnology, contribution from Biotechnology Society of Nigerian Working Group, published by Ruth Ligel, Taylor and Francis, United States of America. A devoted mentor and teacher, Professor Ekundayo has supervised the research work of over 200 undergraduate students, 8 masters and 1 PhD graduate. Awards, honors, and fellowship. Professor Ekundayo's excellence has been recognized through several awards and fellowship, including World Bank's Step B Innovator of Tomorrow Award for screening medicinal plant for antimicrobacterial properties, Third Fund Institutional-Based Research Grant 2012 and 2018, International Center for Genetic Engineering and Biotechnology Fellowship, Sri Lanka, 2015. He is also a fellow of Biotechnology Society of Nigeria, FBSN. Faith, service, and mentorship. A devout Christian, Professor Ekundayo has been deeply involved in faith-based youth mentoring. As an undergraduate, he served as General Coordinator of Deeper Life Campus Fellowship, University of Jos, and later as Region and State Coordinator of Deeper Life Christian Fellowship in Abia State. His passion for youth development inspired him to found the Beacon and Guide Institute, where he serves as president. The organization is devoted to success education and personal development for young people. Personal life. Professor Emmanuel Olufemi Ekundayo is happily married to Mrs. Charity Iroma Ekundayo. Sorry, madam, I was speaking Yoruba to you the other time. I now know why you are looking at me. And their union is blessed with 3 wonderful adult children. Professor, thank you. A round of applause, please. Professor Emmanuel Olufemi Ekundayo remains a distinguished scholar, exemplary administrator, committed mentor, and inspiring model of integrity, diligence, and faith in the academic and scientific community. At this junction, I present to you the 34th inaugural lecturer, Professor Emmanuel Olufemi Ekundayo.
Good evening, ladies and gentlemen. The Vice Chancellor, dear Professor Olayemi Drotimi Akinwumi, Deputy Vice Chancellors, Academic and Administration, Registrar, Bossa, University Librarian, Provost College of Postgraduate Studies, Provost College of Health Sciences, Deans of Faculties, Directors, Head of Departments, distinguished professors and members of Senate, members of the university community and colleagues, esteemed guests, friends, and well-wishers, great FUS students, and my former students, ladies and gentlemen. It gives me great pleasure this evening, and I'm deeply grateful to the Almighty God for the grace He has given me to stand today to deliver the 34th inaugural lecture of this great university. I thank the Almighty God for life, health, an opportunity, and the privilege to contribute to knowledge and human capacity development. I'm grateful, particularly to our visionary Vice Chancellor. I came here just yesterday, and to many of you here, you'll be surprised that when did this one come here and is already giving inaugural lecture. But when I announced it to my former university, they were so surprised because probably it will take me the next 5 years to stand to deliver that lecture because of the way things are done. But I thank God for our Vice Chancellor who has the remarkable gift of spotting and unveiling hidden stars. I cannot go into the details how I come to be standing here, but it takes being under the leadership of such a visionary person who is concerned about the development of everybody. Thank you very much, sir. The testimony of my life is aptly captured in the scripture, Psalm 113, verses 5 to 8. And sometime in 1990, I stood on the podium at the University of Jos, and I read that scripture to them, and I told them that you will hear of me. It says, who is like unto the Lord, our God, who dwelleth on high who humbled himself to behold the things that are in heaven and in the earth. He raised up the poor out of the dust and lifted the needy out of the dunghill, that he may set him with the princes, even the princes of his people. I want to say clearly, I am the poor that God has raised from the dust, and the needy he lifted up out of the dunghill and set with the praises of his people. I give all the glory to the Almighty God. Because when you talk about a humble beginning, I can talk about the humblest of the humble beginning. When we started primary school, there's nobody that will ever think that I will be the first person to become a professor among them. But it is what God has done. May his name be glorified forever. So that's the outline of my lecture today. We're going to briefly look at all that, introduction, biotechnology, the science, so you can see it there. So let's go right into that. I want to say clearly that the word biotechnology has become a catchword around the world. And why I chose this topic, I'll tell you a story. When I was appointed the second time as the acting director of Center for Molecular Bioscience and Biotechnology, my new VC, a mathematician, and we had correspondence and all that. So one day he immediately to me, he said, "Director, what is all this noise about biotechnology?" And I took what he said. I didn't know. So I replied back. Then he replied back to me and said, "No, I didn't mean evil. I just want to understand because I'm hearing about technology everywhere. So I don't want to take it for granted that because you have been hearing about biotechnology that you actually know what it is." And being the first person given the privilege to give this inaugural lecture in my department, I decided to speak about this so that people will be more enlightened about biotechnology and know that it is not like any other subject. Well, if you look at my introduction there, I am professor of medical microbiology and microbial genetics. Why am I not professor of biotechnology? Well, professors of biotechnology are very rare in Nigeria. There are not many in Nigeria because biotechnology as an academic course is a recent thing. When we started, actually, there was no biotechnology. So we have to read other things. So biotechnology is taken from two words, bio and technology. Bio is life in Greek. That's the root word for biology, which means the study of life. And technology is from the Greek word techne, which relates to making things using art or skill. So biotechnology refers to the practical application of biological knowledge to make things that are beneficial to human beings. It is not the same as microbiology, biochemistry, and any of all those things, but you cannot be a biotechnology without knowing any of those things. So as we look at biotechnology, that's where it came from. And the origin of the term biotechnology came from a man, an agricultural engineer called Karoli Erecki, a Hungarian man, who in 1919 wrote a book in German where he described, he said, biotechnology of meat, milk, and fats through large-scale agricultural farming. And that was where the word first came to become used. So after that, biotechnology as a word spread all over the world, and that's where it began. So for Erecki, biotechnology is a method to solve society problems like food shortages and energy. Incidentally, he was appointed the Minister of Food Safety. And he used his knowledge, he was an agricultural engineer. So, and I will tell you later to know that biotechnology can come from various fields. In fact, one of the first persons that made so much contribution to biotechnology in Nigeria, our own late Professor Solomon Ogbe from Kogi State, of blessed memory, was a chemical engineer. But he was the first DG of National Biotechnology Center in Nigeria because of his contribution. So now, looking at definition, maybe I should not bother you too much, but quickly, Biotechnology has been described as the use of organisms or their components in industrial or commercial purposes, aided by genetic manipulation and all that. You can find that in Wordsworth's Dictionary of Science and Technology. And in the United Nations Convention on Biological Diversity, Article 2, biotechnology was defined as any technological application that uses biological system, living organisms or derivative thereof to make or modify products or process for specific use. There are other definitions you can see there. So biotechnology is beyond just a course. It is a tool for practical application to solve human problems. That's how it began. And that's the emphasis I want to make in this lecture, that when you look at biotechnology, it's a multidisciplinary field. And so who is a biotechnologist? Now, but a biotechnology can come from several fields. It could read zoology like I did, or it could start with zoology, go to medical microbiology, could be a botanist, cytogenetics, and all that. And so if you survey professors in Nigeria who are in biotechnology, none of us actually earn a degree in biotechnology, because biotechnology as a course just started recently. And in this university, we are just graduating our second set. So it was around 2004 that one of the past presidents of biotechnology, Professor James Ogbonna, who is now the Vice Chancellor of Medical University, State Medical University in Enugu, who advocated and gathered some people to write the first proposal to NUC that to really benefit from biotechnology, we need to run a course. Because before that time, some people simply changed botany to plant science and biotechnology. Some people added biotechnology to existing program in microbiology or biochemistry. But they look at it and say, we need a real course where people can study biotechnology. So I did not start as a biotechnologist, but a biotechnology is somebody who studies biology, biological sciences, with one distinction, with the aim of developing products and solutions. I can be a microbiology and just do basic science without intention of producing anything. So why I'm a medical microbiologist and a biotechnologist is because I focus on developing products and solutions using biological system. Many people here, including our DVC, I know she's a member of BSN. So many of us, many people here are members of BSN, even though they are in various primary disciplines. So it's a multidisciplinary field, and it draws resources from, you can see there. So as you look about technology, you see there are a lot of fields that contribute to it, and then there are also outputs. Outputs such as, that's where we get a lot of things, vaccines and everything. And I want to tell you, if you have ever eaten gari, or iru, or uba, or ogri, or you have ever taken antibiotics, or you have ever eaten bread, or you have taken beer, or palm wine, then you have been consuming biotechnology products. Because that's where they came from. So, but now so about biotechnology, in a simple term, is a person who is, a biotechnology may be rightly described as a student of 4 biological sciences and a master of research, innovation, and development of products and services that address societal needs. If you are in microbiology, in biochemistry, in other biological sciences, and you are not interested in developing products, you are not interested in innovation, you are just a microbiologist, a biochemist, a zoologist, a botanist. But when you step and you direct your research at trying to solve problems, then, and you learn the tools, then you become a biotechnologist like me. Yes, you become a biotechnologist like me. So thank you very much. Now, let me look at why did I choose this topic? Just to go back. My topic is the science, the products, and the benefits, harnessing indigenous bioresources for national biotechnology. From my citation, you will understand. Well, fate made it that I got myself into herbal medicine research. as a graduate, as an assistant early in life. And from there, I was exposed to, we actually had a traditional practitioner that was employed by University of Jos, and the woman was teaching us, was showing us medicinal plants, telling us the uses, and my role was to run assays. And in the process of that, I was seconded to International Center for Ethnomedicine and Drug Development at Nsukka, which was started by Professor Maurice Iwu, the same Maurice Iwu that you knew, who became INEC chairman. So between 1997 to 1999, I was working in the center as a bioassay manager and microbiologist for screening. We went around Southeast collecting medicinal plants and screening them to see, including screening them for anti-malarial properties and all that. So now the interest is this. Biotechnology has been described as one of the most transformative science or field of science in 21st century because it has application that apply to health, to agriculture, to industry, and to environmental sustainability, issues that are of global interest. And understanding the business aspect of biotechnology is essential for transforming this scientific knowledge that we have into products that generate enterprises and create value for the society. So biotechnology is not just a course. It is something that we need to understand. As we look at Nigeria, we are endowed with a lot of bioresources. When we talk about bioresources, we are talking about plants, microorganisms, and various things that are there. They have a lot of value by themselves. Incidentally, God has made it that microorganisms cause disease, and God has also made microorganisms to be the source of remedy for those diseases. Because through biotechnology, we convert them to vaccine to prevent diseases. Through biotechnology, we convert them to, we derive antibiotics from them. Most of the antibiotics that you use are made by microorganisms. Not only that, from ancient time, our forefathers have relied on medicinal plants to cure diseases. So Africa has a very huge amount of indigenous knowledge about the use of these plants. So unless indigenous bioresources build scientific and economic sovereignty when we properly use them. Biotechnology industry is valued at trillions globally and growing. This is one thing I want to emphasize because, you know, when biotechnology started, we joined the bandwagon like other people, but now they have left us far behind. India, China, and other countries, even South Africa, they are counting already in billions. Big industry, but it's not yet like that in Nigeria. In Nigeria, many times we are just satisfied to start a course without really understanding what is behind it. That's one of the reasons for this, my lecture. Africa is rich in bioresources, but low in value addition. When you look at the gamut of biotechnology, we have an advantage. And that advantage is the fact that we are rich in bioresources. We are rich in microorganisms that are not found anywhere else in the world. It will interest you, there's a company in Abuja, incidentally not a Nigerian company, it's an Indian company. And they have gone to all the local governments in our country to collect microorganisms that they are now using as biofertilizer and selling to people. This company, they came all the way and they went around. When we visited the place, we were saying, now, look, who licensed these people? Who gave them the permission? They just came, they said they are doing research. And they have gone to all the states. They have characterized microorganisms from the soil. And if you ask them, I don't want to use chemical fertilizer, I want to use biofertilizer, they will ask you, which state are you from? You tell them they are going to grow those organisms and give to you. That company has an industry, has a factory already in Casino where they are producing biofertilizer. So what I'm saying is that we have this huge thing that we cannot find anywhere. Before I finish, I will show you something that we discovered during my research, which is unique all over the world. And we put it there, it's there in my name and the name of my student that did it. So because we have this thing, so harnessing biotechnology drives industrial growth, jobs, food, and health security. And globally, This industry is now at the forefront of competitiveness. All major countries in the world have developed biotechnology policy, bioeconomy policy. Nigeria developed one, but as you know, in Nigeria, we have very, very good policies that we play with. Other people are doing so. When we go to conferences and we say, every country develop your biotechnology policy, they call us together, we develop beautiful one, and the thing is lying there somewhere in somebody's store. Now let's go on to what we are really going to talk about, the science. The science. The science of biotechnology. I want to tell you that biotechnology is of ancient origin, but the science is of recent matter, just recently. From the ancient time, our forefathers who began to look at plants and animals and domesticated them began the process of biotechnology. So biotechnology began with domesticating animals and also began with when ancient people, they were looking for a way to preserve their food, and they made bottles from skin of animals, from the bladders of animals, and kept their milk inside those bottles. And because those tissues contain an enzyme, that enzyme acted on their milk, and their milk turned to something else. And they felt this tastes better, the milk doesn't spoil, and that was how they learned, without knowing the basis of what they were doing. Gradually, they learned that if you expose milk to the atmosphere for some time, it takes on a different flavor, and that milk will not spoil. They stumble into yogurt, stumble into cheese, probably the way they stumble into iru, and stumble to ogiri and uba that are delicacies around the world. These are all practice of biotechnology before even the science was known. As we look at various stages of development of science, we can identify 3 stages: ancient biotechnology, classical biotechnology, and modern biotechnology. And the chart you see there is a depiction that somebody has made and said, and they try to outline. And at the top of that chart is where will it lead to? We don't even know. Because today, biotechnology is producing some fearful thing, which is what some people know about biotechnology. They're afraid of it. But I want to tell you that what they're afraid of is just an iceberg of what is in biotechnology. The real biotechnology does not need to scare anybody. It is the solution to so many problems in our life. So what the people are hyping in the news and telling us this and this is just an infinitesimal part of what biotechnology is. So other people have classified biotechnology as traditional biotechnology or modern biotechnology. Now, traditional biotechnology covers all those things that began after 1800 to the present time. And what biotechnology has done today is to refine the old practices, understand the basis, and make use of them. So we still practice biotechnology. Like me, I do research on medicinal plants, which ancient people have used, but I'm not doing the kind of thing they are doing. I want to know precisely what does this medicinal plant do? What does it act on? Which microorganisms can it kill? But the ancient man goes to the bush and by empirical, I say, if I gather this and gather this, it will help. And it does help, but sometimes it does also harm them. So for us as biotechnology, we rely on this indigenous knowledge, but refine it today with the tool that we have to know how to make it safe and very useful to us. Traditional biotechnology covers all the techniques that have been used from ancient times. It includes fermentation, used in the production of fermented foods, such as gari, ogi, akamu, as you like, iru, ogri, uba, gari, fufu, kunu. All those things are the product of traditional biotechnology. And the people that still do it in the traditional way, they just do it, they grind their cassava, cover it, and they know that after 2 or 3 days, something will happen. They don't know what is happening, but I know what is happening. Then people are making kunu, and sometimes the kunu goes bad. What we do in biotechnology is to find out how do we identify what is working in this kunu, because we have understood that kunu is produced by actions of several microorganisms. And that's why you have different tastes. But now we are now looking at it that we can, as I've come here, some of my students already, my final year student, we did a lot of work. We went to behind Fililia campus, went to collect milk from the Fulani people, and we're trying to identify the things that make their kunu to be what it is. And I want to tell you that the first student I had first class in my department happened to be my student when I was visiting here. And then when I told him what we are going to do, the department said, "Hey, how will he be able to do this?" And I told them there's no need. But it costs a lot of money, because at the end of the day, I had to take some part of that research to my lab in Umaya to do. But to God be the glory. That student is now serving, and he has already written all that paper and sent a draft to me. So that's what we are doing, trying to look at this local indigenous knowledge and see how we can make them better. Let me just go quickly now. Thank you. Now, modern biotechnology is about manipulating a little chemical that we call deoxyribonucleic acid. When I teach my students microbial genetics, I will tell them that all of us are chemicals. That's what I start with. All of them will be looking at me. And I will prove to them that all of us are chemicals. At the end of the day, I will ask you, are you a chemical or not? So, Vice Chancellor, sir, every one of us here, elephants and plants and everything, we have one thing that unites all of us together, including E. coli, a microscopic organism that we cannot see. That thing is called DNA. It is what determines how tall I am, my complexion, and everything about me. So before 1940, that molecule was known, but nobody knew what it was doing. But by 1940, some people stumbled on by accident the fact that because they had known DNA before that, but they never knew this, they thought this thing was too small to determine the genetics of human beings. But by 1940, it was established. And by 1953, Francis and Crick, incidentally, James Crick just died a few days ago at the age of 97. They discovered one of the things that revolutionized the whole biotechnology. In fact, that's just the beginning. They discovered the structure of DNA. And that structure of DNA explains how that little molecule, you can see, they propose what they call, if you look at what you see, what they call double helix. And so they tell us that this double helix, this DNA molecule, is responsible for everything that happened in our life. The second diagram there, you see DNA, deoxyribonucleic acid, and then RNA, that is ribonucleic acid, a kind of another tribe of DNA, work together to produce protein that controls everything in our body. We call that central dogma. And that's the basis of the whole thing about the science of biotechnology. But like I said, everything about biotechnology is not just DNA. We also make use of the cell, host cells themselves. So a biotechnology studies the cells, animal cell, microbial cell, plant cell, to understand the processes. And when we understand the processes, we apply those processes. For example, when the moment the DNA structure was known, by 1976, some people discovered that you can actually take a DNA from a frog and join it to a DNA from a bacterium, and the bacterium will not recognize whether it is from it or it's from toad. And that's what the beginning of what we call genetic engineering. That you can take, that's the basis of what all that we are hearing today, you can engineer an organism. And by 1980, a company called Gentech actually took the pancreas, insulin. Before 1980, diabetes patients can only get insulin by extraction from pigs, from the pancreas of pigs. It was in limited supply. But by 1980, when scientists have discovered that there is a thread of life that joined all of us together, DNA. So they took the gene from human pancreas and put it in E. coli, a microscopic organism that is the guinea pig of laboratories and the most friendly of all because it's in our stomach and all over our body. So they put it there, and that microorganism faithfully produced insulin, which was called humulin. So if you have ever taken insulin, or you have anybody taking and you have bought humulin, humulin is actually insulin produced by E. coli, genetically modified organism that became a huge industry today. So with that, there was no scarcity of insulin anymore. That's the power of biotechnology. That's what biotechnology does. Trying to provide solution. Then recently, we have heard about genetically modified corn, which is generating so much noise in Nigeria. And for some of us that know the basis, somebody will say, is genetically modified organism safe? The truth is that nature has always modified organisms. What biotechnology does is just to make it faster. In time, even you human beings, in time, some of us undergo changes. It's gradual. But as biotechnology, we can make it fast. What will happen in 1,000 years can be reduced to a matter of hours. And that's the difference. But some people say, oh, the technique you are using is not safe and this. There's no drug as safe as GMO approved products. I said there's no drug. There's no medicine that you are taking that is as safe as a product that has been approved. Because we, biotechnology, we are not making product to kill ourselves. We are making product to make work better. I think you clap for us. So thank you. We play with word when we talk about biotechnology. We talk about rainbow or color-coded biotechnology. So we talk about red biotechnology, which is medical biotechnology. Biotechnology applied to healthcare. Then we talk about green, which is agricultural biotechnology. White biotechnology is industrial biotechnology, and gray biotechnology has to do with environmental. These are applications. And of recent, we also come across what we call blue biotechnology that is going to the marine. We now discover that there are so many resources in the ocean. So there's another thing we now call blue biotechnology. And of recent, I'm also hearing there are other shades of these colors. So just tell us that we just make ourselves happy. So for me, I'll be carrying the red flag because I'm a medical biotechnologist. I told us that you could be a basic biological scientist. What really makes you to become a biotechnology is when you learn the tools that make you to become innovative and product development oriented. And so we have a lot of tools that I'll just run through quickly because of my time. So we have tools and techniques that biotechnology use. And for us, for departments and places that are running these are the tools that we need if we are going to practice biotechnology as it's supposed to be practiced. We have cell and tissue culture techniques. This is where we culture cells. Cells, we are all, I said all of us have DNA, but all of us are also made of cells. Some organisms like bacteria, they are just one single cell, their whole life. Mother and father and children, only one single cell. Human beings will have billions of cells that are specialized, but all of us are made of cells. So before we can even study DNA, we must culture the cell. We must be able to take the cell out of their natural environment, put in artificial environment so that we can manipulate them. So we use cell and tissue culture techniques. And the simplest is culturing microorganisms. They are very fast. Like E. coli I was telling you about can grow. E. coli can reproduce in 20 minutes. And recently, 3-4 days ago, I was reading, there's another organism that has just been discovered that can even reproduce in 10 minutes. And the student that found that in the, I think it's University of California, they are on their way to another Nobel Prize because now they have formed a company and the thing is going to be used. So there are things undiscovered. When I was graduating from federal school, one of my students said, Emma, don't worry yourself. Just go and find a lab and work. All the things to be discovered have been discovered. And I told him, I said, don't worry. In the future, you will know that what you are saying is not true. And wherever he is today, he will understand. So there are so many things to be discovered. In fact, we are told we have not discovered more than 3% of the microorganisms that are in the world. And incidentally, let me interest you. As you are sitting down here, your body is made of 50% of cells that are not part of your body. Does it sound interesting? As we are here now, we have what we call natural microbiomes. And these are cells of other things, but our body is made of 50% of that. So when you say, me, well, you should understand what you are talking about. But God has made it so. So they are part and parcel of us anyway. But we have now discovered that those organisms, they are so important to us. They help us to digest our food. They help our brain. They help our blood pressure. They help everything. That's another aspect of biotechnology we are just discovering. And now people are coming to Africa to take, well, sorry to say this, maybe you don't like it, but when I teach my students, I say, there's nothing to be, when we talk about fecal matter, some people say, But no matter how gaily you dress, the fecal matter are inside your stomach. They only become repulsive when they come outside. So some people are coming to Africa to collect our poo-poo because they have now discovered that we have some things, we have some microorganisms that make us to be more resistant to cancer and so many things and all that. So they are now coming here to find out. But if we don't know, we sit down here and sometimes they will tell us that they are collaborating with us. We gather all our specimens and send to them. Then tomorrow they will produce the product and send back to you. When I was in Interseed, we were working on bitter cola. You know, if you have heard about Professor Morris, who, you know, he talks about bitter cola a lot. We were working about bitter cola and we collected tons of bitter cola. We were refining them and all that. Eventually, he went abroad and made a product and came back to us and said, this product is too expensive for Africans. That's just the truth. But but our forefathers have been eating bitter cola, and you think they are just doing tradition. But very powerful antiviral products. So we did all those things that was part of the research. Eventually, they took it abroad, sold it to a company that produces and brought it back. Then he showed us a sample and said, this drug is too expensive for Nigeria to buy. So we have genetic engineering and bioprocessing analytical tools. Let me go quickly because I will elaborate on this more later. So let's go now to the products. I've just mentioned a few things about the products. The products of biotechnology includes vaccines, humulin, like I talked about, insulin, genetically modified corn that you do not need to spray with insecticide. Because some years ago, some people discovered a bacterium called Bacillus thuringiensis in the ground, soil microorganism. But this soil microorganism has a characteristic that when insect eats and pick up the spore will germinate inside that insect and produce a toxin that will kill the insect. So some people now say, oh, what if we put the gene inside plants? So they succeeded, they put the gene inside cotton. So we have BT cotton today. We have BT corn, that these insects that come and lay egg, when they lay egg, when those larvae hatches and they eat part of that tissue, they just die off. So that reduces the amount of chemical we are using in our environment. We already have that in Nigeria. That has been approved in Nigeria. So today we have BT cotton, and nobody bothered about BT cotton so much, but recently when we talk about BT corn, maize, there was an uproar. They said they have come to put poison in our food. Well, sorry for you. If you eat popcorn from these popular places, you have been eating that one for a long time and you have not died. So that's good news for you. If you have been eating the exotic biscuits, well, it happened that you have been eating a lot of BT corn. So it's not killing anybody. We have these products and we can divide them along the line. of the various fields. We have medical products, mainly vaccines, antibiotics, diagnostic kits that we use for testing for diseases and all that. We also have agricultural products, which include transgenic crops, like I've said, like BT cotton, BT corn, and all that. To date, and officially, we only have 3 approved in Nigeria. Some people will tell you that, this apple that doesn't have seed is GM. There's no GMO apple in Nigeria. There's no GMO anything apart from BT cotton, Taylor maize, and cowpea. These are the officially known released, which were tested over 15 years before they were released. Then we have industrial products, which include enzymes that are produced, enzymes that are used in industry, amylase and all those. Then we also have environmental products. These are consortium of microorganisms that can clean up polluted environments. Bioremediation, areas that have been polluted by oil, we can actually gather microorganisms to help us clean up that environment. They break down those things and remove that. Then we have other emerging frontiers of biotechnology. Let me now quickly talk about the development process. And why I'm talking about this is because of where I want us to go. Now, the development process is a kind of cyclic thing that runs through 7 areas. It begins with basic research and discovery. The thing that we are commonly doing, because we do not have capacity to move beyond that. A lot of us, I did a lot of that because after doing it, I will come to a crossroad, I cannot proceed because it's too expensive. So thank you. I'm a preacher, so I can preach for a long time without water. Thank you very much. Somebody told my wife, we better prepare water. She said, my husband is a preacher. Thank you very much, I appreciate it. So we have basic research, which is what we have been doing. But money is not in the basic research. It is necessary, but there's no money there. That's not where the money is. Unfortunately, that's where we are stuck. And the idea behind my lecture is that we want to move beyond that. So we have 7 steps, basic research. Then we have, we also have, we have basic research. We have applied research. That's where you have, when you have done the basic, you now want to try. Like research I did, I will describe about after years of doing medicinal screening. I said, how do we go from this place? And then an idea will come to us and we'll start doing that. So when you look at a promising discovery or a promising innovation, and you now say, where can I apply this? Where can we use this? That is applied research. Then we also have product optimization and preclinical development. If you are dealing with drug, if you are not dealing with drug, there are other tests. At this stage, you have selected a product, but you are now trying to optimize it. You are now trying to see which is the best way. When penicillin was first discovered, it was Penicillium notatum that was producing it. But in the process of optimization, they now discovered there is another species that can even produce more and better, which is what is used industrially. So when we do research, basic research, and we discover something, we say, okay, let us go around. Can we find other species of microorganism, or can we find another plant that can even produce this more? That's optimization in the process of development. Then we have scale up and process development. At this stage, you are now looking at, you have come to a critical point where you are looking for a partner in the industry. You have discovered, you have done the basic research, but now you need scale up, you need money. And that requires working with industry, collaborating with industry. That is very, very low in Nigeria today because the industry seems not to be interested. But abroad, the COVID-19 we are talking about, research has gone on it for years. And then when the time came, they gave them to companies, to Johnson & Johnson, and to all those people that produce them. But this research were done at Hopkins, at various universities. So this is where we need, and let me say something. If we do not have companies that we can lend to, why can't we start our own companies? I will tell you at the end of the day how I came about the idea. So instead of just saying, nobody is doing, why can't you start? These biotechnology students we are producing, why can't they start their own company and grow it? Just like they are doing in digital things today. And when you are brought to a point, you will see company that will come and buy it. But no company is going to be, no company will just run to you when you have nothing to offer. So we can start our own company. Biotechnology today, you can start small. And when you get to a point, you have a product that you can sell, like people are selling digital products. Incidentally, there's also a digital product in biotechnology. I will show you that later. Then commercialization and marketing development. That is the time that you give your product to a company to market for you. Thank you very much. I will quickly run through my contribution to biotechnology research and capacity development. Based on my background as a medical microbiology and microbial genetics, my research has focused on the characterization of antimicrobial resistance. Now, antimicrobial resistance is a big issue in the world, and it simply means that we are now experiencing a situation whereby a doctor prescribes an antibiotic for you based on what he has seen, and then you take it and it doesn't work. You know, in the year past, we had a lot. Okay, thank you. We had a lot about chloroquine, but today you see that no doctors prescribe chloroquine. Why? Because chloroquine is no longer effective. Plasmodium, malaria parasite, had developed resistance to the drug, mainly because of abuse, and so many things like that. So antimicrobial resistance is a big problem in the whole world, and we need to have data on them. So I started my research by trying to collect information on the resistance of common pathogens, bacteria that cause disease in our environment. We go to clinic, we go to places and collect specimen, culture them, and try to test them against this bacteria. There's a simple device. There's a simple device. That thing you are looking there is called antibiotic susceptibility disc. These are just filter paper that are impregnated with antibiotics of various kinds. So when you go to the clinic, when you go to a lab and they say, go and do sensitivity test, they culture and find out isolated organisms and test them against a battery of antibiotics, and then send the result to the doctor to know which one to use. So when I got to Umudike in 2005, I discovered that because these antibiotics, they were very simple thing, but we are importing them. And the difference between the imported product and so some people in Nigeria started manufacturing them. But the experience as a medical lab scientist, I have worked on the bench, the experience I had was that some of these things, they give abnormal results. So the first project I did in Umudike was to collect these locally produced antibiotics and try to test them against a foreign one. And then we did that, and that paper generated so much interest that other people have done a lot of work because of that thing that we did. And we discovered that those locally produced antibiotics, they are very simple to produce. Actually, I developed them and I've used them. I've used the one I produced myself to do my research for a long time. Sometimes I still buy. You can imagine where I have to spend 36,000 to buy just filter paper that I can make in the lab. But to publish internationally, you have to show that you have a reference. So, but we produced that. So when we did that test, we discovered that those ones produced in Nigeria, they were not standardized. And they were not standardized in the sense that if you use that and you send to a doctor and doctor follow that to prescribe drug, the person will not be healed. And I was discussing with a doctor and he said, tell me, why do they send me result and after I treat the patient, they don't get healed? And I said, what kind of antibiotic discs are they using? He said, it doesn't know. I said, probably they are using these locally made ones that are not standardized because they do not control them. And the process of controlling them is very simple. We do that in my lab, and I have one of not my student now, was my student before, but we are guru in producing that now. We produce for ourselves and use, and then it's as good as the foreign one. Thank you. In fact, we have worked on the we have worked to try to see how we can design it so that you look different from any other one so that we can begin to sell. But, you need money to commercialize things. That's the problem. So that is just the table is just showing you the doctors that are here, also people that are familiar. These are common antibiotics that are usually used, prescribed in Nigeria. So we concentrated on them because the idea is that WHO and people all over the world, they say, select antibiotics that are used in the country and test them. So in the process, we gather a lot of data, a lot of information, very useful, that contribute to the global database, because all over the world, they want to know what is happening in Nigeria, what is happening there. And particularly, we concentrated on testing again Staph aureus, Escherichia coli, and Plasmodium aeruginosa. These are common pathogens that cause disease in Nigeria. So my work has contributed a lot. Then some time ago, we came across that a lot of people were getting sick from salmonella. Salmonella is what caused typhoid fever. Now they have given me time, I'm writing on soon. Salmonella is what causes typhoid fever. And some people say, where do I get typhoid from? You buy egg. And you buy egg and you bring egg to your house and you think you are safe. So we collected eggs in Atumudike from farms and tried to see whether we isolate salmonella from them. And we actually did. And if you look at that chart there, you see that those blue something represents those organisms that were susceptible to the antibiotics. And many of them were not susceptible. These are from eggs. And because chickens have, they can contaminate egg in the process of laying the egg. And when the egg hardens, the salmonella will be inside the egg. Please, I didn't say you should not eat egg. You understand me? You have been eating egg and you are okay, so continue to eat egg. I'm just telling you that these are the kind of things we found out, and very, very important information. So, but it is not that salmonella is in the egg that is our concern. It is the level of resistance to antibiotics that was our concern. And we published that, and a lot of people started to say, oh, so this is what is going on in our environment. Then we also did another series of tests. We look at antibiotic, because when people go to go and test, when you go to the lab, they say, go and do malaria and typhoid. And they do what they call wither test. That wither test is just a test that is so insensitive and all that. And sometimes they tell you have typhoid, you don't have typhoid. Sometimes they say you don't have typhoid and you actually have it. But the most sensitive test is to do what we call stool culture, which takes about 3 or 4 days. And people are not patient to wait. They just say, no, do it now, do it now. So I am one of my students here, who is almost, if not because of some problem we are familiar with, as just like another, he will have defended his PhD. So we decided to culture, do what people are not doing. We collected people's stools and urine and isolated salmonella, and then tried to look at. So if you look at this chart, you see all the blue, that is sensitive. And all those red ones, they are what? Resistant. That's what we found. That this organism, so when you go the prescribed ciprofloxacin empirically without testing you take it, doesn't work. You come back and say, doctor, the thing has not gone. It has not gone because the organism is resistant and the lab is not willing to do tests for it. So we let that then. I started going beyond just doing screening. We wanted to do molecular screening. We wanted to identify the genes that are responsible for this. So with this series of work, you can see there, those are the publications. We look at the genes that are responsible for antibiotic resistance in various microorganisms in the south, and southeast. Then I want to talk about this very, very interesting thing. This thing you are looking at on the board is actually in the process of collecting specimens from poultry farms. Like the inaugural lecture last week told us how much it costs how much it takes them to be able to send sample abroad. This thing cannot actually be done in Nigeria. So we spend a lot of money and we cannot do much. So we collaborated with somebody in Lagos that used to collect a specimen to send to South Africa. But in the process, we discovered that when we did this and they sent the result back to us, we discovered that this organism, Klebsiella and Proteus mirabilis, are so unique all over the world. They have not been seen. They're only in our sample. So Now, if you look at it, if not because it's tiny, if you look at it, you see my name is there and the name of the student. We uploaded to NCBI, that is National Center for Biotechnology. The importance of that is this: in the future and all over the world, anybody that wants to detect this organism can always go to this, our sequence. What you are seeing there, written TTTG, those are DNA sequences. You can design the agonistic test that will be able to help you. So in the future, for example, I can start to say in Lokoja, this thing that we saw in Numaya, I see in Lokoja, then I'll just design primer and start to test. So that is there in our name. And if we have more money, we can do more than that. But I know that costs us a lot of money. And the student herself was not, but we did it. Thank God. Please permit me to, I will jump to this. Now, after screening medicinal plant for a long time, I reasoned, what can we do with this? We discovered that all over the world, poultry farms are the epicenter of antimicrobial resistance, because farmers are using antibiotics that human beings are using to treat their animals, to prophylactically. And because of that, this place, so all over the world, some countries in Europe, America, they have banned the use of antibiotics in poultry. But in Nigeria, we're still doing it. But we cannot take something away without replacing it. So we reasoned and we wrote a proposal for TED Fund and got IBR TED Fund to try some medicinal plants as replacement for antibiotics in poultry farm. And we got wonderful results. And so it was very interesting. In fact, a professor of animal production saw our chicken 8 weeks and said, I can't believe, what did you do? You say he did not give this animal. We said, no, look at the medicinal plant we gave them. And so that's something we are developing, because it's very, very important that we can actually find our environment from medicinal plants that our forefathers have been using. And also when we are doing this research, I discovered that Fulani people in the north, a lot of them also use a lot of medicinal plants to treat their poultry, their local chicken. And we can do that. Ladies and gentlemen, let me jump quickly because I must talk about this. Now, business. Let's go to business. And this business, I'll just summarize. Biotechnology is not about just book, it's about business. In 2015, I got a fellowship to go to Sri Lanka on international genetic engineering and biotechnology. And they took us for a training. And then when we got there, they took us to a company which was called Syngene Biotech. They told us the history that somebody went abroad to study from Sri Lanka. When he came back, he discovered that he could not get material to continue his work. So he went to the University of Colombo and got in touch with professors and said, what can we do, you professors, what can we do so that we can produce this thing in Nigeria that will help our education and help our healthcare? So they formed a company they call Syngene. And that Syngene company, they started producing things. So when I went there, there were 4 Nigerians. Somebody came from Malaysia, was a PhD student, and 3 of us went from Nigeria. They showed us everything, took us around, and showed us what they were doing. And in the process, we learned that with collaboration, we can do a lot of things. So that is me in that workshop in Syngene Biotech. In Syngene Biotech, we discovered that a private hospital, collaborated with people in the academy, and they formed a company and started manufacturing reagents and all that. In 2019, when COVID-19 came, this Syngene company became so pivotal. Okay, thank you. Became so pivotal in the control of COVID-19 in Sri Lanka. While in Nigeria, we are running helter-skelter looking for where we diagnose COVID. What am I saying? You see, with simple collaboration with university and hospital and private people, we can kickstart what we call biotechnology business. The book is in your hand, you can read through it. This business is what we can start. And I want to say that here, the passion, what brought me here actually, what brought me to Federal University Lokoja was because of the vision I had. When after being a director of a center for 5 years, I said, it's time to go back to my state, where I knew biotechnology were going to start. And thank God, glory be to God, I met the VC, discussed with him. He said, you can come here on sabbatical. And we made an arrangement. Eventually I came, we started the biotechnology department together. And today I am here. And my vision, because inaugural lecture is also to tell you what I intend doing. My vision is that we should start an institute of biotechnology here. That institute is beyond a department. It will enable other people in other departments to collaborate and enable us to collaborate with industry. The kind of thing that I saw in Sri Lanka. And it can be done. I couldn't go to detail, but in my lecture, there are low-hanging things that we can do. Biotechnology is very, very expensive, but there is a place we can start from. From the academy, we can start to look at things that we can do that when we bring it to a place, we will be able to offer something to the industry. Nigeria always say there's poor connection between industry and academy. The reason is because we are waiting for industries to bring their money to us. It doesn't work that way. But when we have something to offer to them that has potential, and biotechnology is actually that kind of thing. It's huge money all over the world. If we don't do that, we will say we have Department of Biotechnology, but it will be like any other department where nothing will be happening. So, ladies and gentlemen, I thank you very much for listening to me. Institute of Biotechnology, Federal University Lokoja is my dream. And with that, we'll be able to realize not only the science, we'll be able to develop the products, and we'll be able to do the business as it should be done. Thank you very much.
To be the best, number one and nothing less. Lead me to my destiny.
Some can we rise? Can we get a sound louder?
I believe I know I can count on me. So stand up.
All the champions, stand up, stand up, stand up. For the champions, stand up. For the champions, stand up, stand up. For the champions,
Thank you very much. Can you please put your hands together, make it very loud for Professor Ekundayo? Can you make the ovation really, 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 really loud? Thank you very much, the registrar.
Distinguished ladies and gentlemen, what an enlightening and inspiring inaugural lecture we have just witnessed. Can we have another round of applause for the inaugural lecturer? The lecturer has brilliantly demonstrated how science and innovation can translate our nation's rich bioresources into real economic value. Sir, we thank you for this inspiring lecture, and we really appreciate you for that. May I, at this junction, invite our Vice Chancellor, Professor Olayemi Akinwumi, to appreciate our guest and also perform other functions as scheduled. Vice Chancellor, sir.
Thank you, the registrar. Please, another round of applause for our lecturer. At the last inaugural lecture, I said, our inaugural lecture is getting better every lecture. And today we have listened to a classical lecture. And I think we are now better informed about the world of biotechnology. As he was giving his lecture, I was also telling myself that I think I'm also about technology. On a serious note, I'll give you an example. When I was doing my PhD work, I'm a historian, and my work is on intergroup relation. And I was examining intergroup relation in Bogu. Bogu is in Nigeria and Republic of Benin. They are very proud set of people. When you ask them a question, the first thing they will tell you is that we are the only ethnic group in Nigeria that the Fulani did not capture. It's not relevant to the question you're asking. No. So I was forced to ask, why you people always say this and what is the secret? They told me that they are poisonous arrows. Poisonous arrows. And when I was doing my PhD, it was during the Gulf War, when America accused Saddam Hussein of using biological chemical warfare. When I asked this question, they went and brought some leaves and showed me some leaves. And I brought it to University of Ilorin. I was teaching University of Ilorin. And they examined it and they saw that the leaf contains some toxins. So it was that information that led me to write a paper, and I titled the paper, Biological Chemical Warfare in Pre-Colonial Nigeria. Yeah. Yes. And it was actually the paper a professor saw in Germany that led to my invitation to Germany. And today I'm grateful to God for the exposure. So when he was talking using plant and to technology, I think I'm a biotechnologist. So I think I'm entitled to register as a biochemist. Yes, sir. Yeah, about technologies. You know, it's because we don't take research very serious. When I was invited to Germany that time, another professor from, I guess, Unsuka, but he did not come back to Nigeria. He ran away to United States. He was also invited. And both of us, we met at the university there with our different plants. I went with my own plant. He came with his own plant also. It's in pharmaceutical something. And you can see in that place, all of us were in the Institute of Anthropology. They call it ethnology. It's a purely humanity program. We are there. You can see how multidisciplinary something is taking place in this country. But he shared, normally we sit together and share jokes. And he told me about his plant one day. And I think the lecturer can enlighten us. He said, this plant in Igbo land, when they want to go and plug this plant, they do it middle of the night. And they are always naked when they go to plug the plant. So what he did was, he went naked, plugged the plant, put it aside. The second day, he went fully dressed and plugged the plant. Then he went one afternoon also, plugged the plant, and he did this research work. Very interesting. He said, those who that it is the same quantity or quality that is in the plant for those who plug it naked and those who are, it is just to mystify the plant. That is one thing that he said, and I started laughing. Then the second one that has to do with the timing. He said in the afternoon, the sun is too much. The sun will have sapped some of the things that are ingredients. You know, I'm not a scientist, so I don't know the term. That will have sapped some of the ingredients, and in the night, it contain more ingredients than you can see how science is going, and you can see how people are. But the good thing about the lecture today is, it's a pastor, and generally when you talk about something, people will be thinking that you are into fetish, you are a juju person. We are making a lot of mistakes. You understand? We are making a lot of mistakes. And I think there is the need to do more research into this. I'll give you another example. I was always having typhoid sometimes ago. Somebody came to meet me in my house in Kefi. I said, I hope this typhoid will not kill me. The person said no. And he said, you mentioned egg. He said I should go to the market and buy this local egg, not this agric egg. Just one, you know, they normally lay just one or two or something like that. Then we brought the local egg and he broke it inside the glass cup and add lime, mix it together, and I took it. You won't believe it, typhoid disappear. Typhoid disappear. But when you tell people, they will say, no, no, no. That is what I've been using to treat typhoid. The second one that he gave me, it was also a leaf. He said, but I have not done the research about it, but I stopped using that leaf because the time you use it, just use it in the night so that you just excrete everything away. You understand? But what he said was, when I take it, I boil it in water, and when it is boiled, you make garri out of it, eba, and you eat it with without meat. You eat it with whether pepper stew or something without meat. And we try it one night. Throughout that night, I was just going to the toilet. but when I woke up, I became very light. It's clear this something. But there's a need for us to go into this, and I think the biotechnologies can assist us to do some of these things. So we are really grateful for this wonderful lecture. And I think I said I'm better informed now. And if the opportunity arises to start what you said you want to start, we will give you the support to do it. And just a matter of writing a proposal for us to take it to Senate to discuss and to approve. I want to thank you again. It has been a wonderful lecture. I want to appreciate the following personalities that are here with us. Lieutenant Colonel Simon Carr, who is representing Commander 12th Brigade, Brigadier General CD. Yeah, you are welcome. Lieutenant Colonel Simon Carr is representing. We have Barrister Otowu Gabriel, is the OC Legal and Prosecution, representing the Commissioner of Police, Bello. You are welcome, sir. We have Dr. Leonard Nduka Okonkwo. He came all the way from Umaya. He's a fellow of pediatric surgery. Please, where are you, sir? Yes, sir. You are welcome, sir. We are welcome, sir. We have Dr. Paulinos Obina, also is here with us. You are welcome, sir. You are welcome. We also have Dr. Iyu Ewa. He's an associate professor from the former university that he came from. Yeah, thank you. You are welcome, sir. Thank you, sir. Engineer Tony Ikwagu. Yes, sir. You are welcome, sir. Thank you, sir. We have Owotogbe Iyo, a family member. Lovett Okechukwu, Ikwagu Onye Kashi, Joy Shoma Ikwagu. You are welcome, ma. Mark Iruoma. You are welcome. Dr. Iberedi Ikwagu. You are welcome, sir. You are welcome, ma. And we have some people joining us online who are online. Yeah, we have people, Emmanuel Ayonsi. We have Chibuzo Uzogu. We have Professor Shola Omodamiru. We have Samuel Uka. We have Ali Asana. Ukwa. So we have Patrick Audu. We have Elizabeth Okonko. We have Emmanuel Olaofe. We have James Anyinson. We have Clement Yinaya. We have Waniku Goodness Baji. We have Uche Deborah. We want to appreciate all these people for their interest in our program, our inaugural lecture. As we have told, this is the 34th inaugural lecture, and 32 under this administration. I want to thank the I want to thank this, the inaugural committee, the inaugural lecture committee. I want to thank you for the good work you are doing. But there is a mistake there, and I think it should be corrected. We only have, for those who have delivered lectures so far, just have 29 here, it's 34. I think something went wrong somewhere. It should be corrected. And this is as tradition. He has spoken, and he has spoken well, and is therefore the charge and acquitted. Congratulations. Please, a round of applause for him. Thank you, please, sir. Please, you can step forward here. The family can join you, please. Yeah, let me invite Lieutenant Colonel Simon Carr to step forward, please. And Barrister Ukuru Gabriel to also step forward. Yeah. Please step forward, please. Lieutenant Colonel, please. You assist us to decorating with this.
Congratulations for this inaugural lecture. On behalf of my Commander, Brigadier General KUCD, I decorate with this.
Yeah. Yes, sir. Please, on behalf of the university.
Congratulations, sir. On behalf of my Commissioner of Police, CP Bello Nasir Kankarafi, CP Kogi State, I present to you this certificate. Congrats.
Thank you, sir. Please, I want to invite Dr. Leonard Nduka Okonkwo to step forward, please.
Be the best. Number one, nothing less. Leave me
Wow, this is beautiful.
the
So on behalf of myself and family, I want to congratulate you today on this beautiful inaugural lecture, the 34th one of this great university. And I present to you this gift. Congratulations, sir.
for the champion.
Thank you, sir. I'm just told that my pastor is around, Pastor Adefila. Please, sir. Please, where are you, sir? Please, can you step forward for a handshake with the subject, sir? Please, a round of applause for Pastor Adefila.
Here we go. Here we go. Now it's just we want to. It's just life, that's how it is. Because we have our strengths and weaknesses. Oh, I have vision, can't you see? I'm on the move, make way for me. Stand up for the champion, stand up.
I asked my pastor to pray for him. We go to the same church together.
Praise the Lord. It's a wonderful thing to be a member of Deeper Life Bible Church. The Vice Chancellor is a member of Deeper Life Bible Church. And in the last few months, this is the third inaugural lecture by members of Deeper Life Bible Church. And I want to assure you, you are part of Deeper Life Bible Church, you are a great person. The devil will be running away from you. The enemy will be running away from you. Success will be your portion. Victory will be your portion. And the name of the Lord will be glorified in your life. Heavenly Father, we thank you very much because you are good. There is none like unto you in heaven and on earth. Thank you for what you have done today. We thank you because it is your doing and it is marvelous in our sight. And you said in your word that we should rejoice with those who are rejoicing. We are rejoicing with our beloved brother today. And Father, we pray greater things will come in Jesus' name. And for all who are here today witnessing this good occasion, good things will come into your life. Because whatsoever you sow, that is what you will reap. As because you are sowing appreciation for what the Lord is doing for this brother, in your own life also, we shall appreciate God. And the name of the Lord will be glorified and be exalted. And Vice Chancellor, more glorious things will happen even before you leave this campus in the name of Jesus. The Lord will be with you. The Lord will stand by you. The Lord will not leave you alone. No weapon fashioned against you will prosper. And every tongue that will want to rise up against you in judgment, God will condemn. It is well with you. Thank you, Heavenly Father, for this day. For in Jesus' name we have prayed.
Thank you, sir. Another round of applause. Thank you, the registrar.
Mr. Vice Chancellor, sir, thank you very much. So I think we have come to the end of today's inaugural lecture, but we have some few announcements which the University PRO will make.
Thank you, sir. All right, distinguished guests, ladies and gentlemen, thank you for always joining us. We're very grateful to have you in our 34th inaugural lecture. For our very distinguished VVIPs up front, the distinguished guests from Umuahia, Mudike, and all other very special dignitaries, you will follow the procession that will be led by the Vice Chancellor. Just follow them from a distance, and they will lead you to a place where the Vice Chancellor will treat you to a soft refreshment. And for the rest of us, please remain seated. You'll be attended to right in this hall. Thank you again. May we now rise for the national anthem and the university anthem, 1 stanzas each. Thank you. The national anthem.
Pride and tongue may differ, in brotherhood we stand. My children's all and proud to serve our sovereign motherland.
Greatest FUL and the greatest FUL. Greatest FUL.
Thank you very much. Please remain standing as the procession exits the hall in reverse order. Please, a quick reminder. All are very important dignitaries, professors, deans, and directors. Join the Vice Chancellor's procession to where you will be treated to a soft refreshment. Please, the rest of us, you may want to remain here where we will be attended to. I'm sure you know how these things happen. It's in levels and categories, and so we belong here while the procession goes to the conference room. Once again, thank you very much and good night.